Next slide, please. Next slide. No. The slide before that, Dr. Akpa. Okay. The clinical importance of autoimmunity in transfusion. Why do we need to study autoimmunity? So that is what this slide is talking about. For instance, we are studying it so that to know which antibody or immunoglobulin is responsible for warm auto antibodies and how can they be detected? Which one is responsible for cold auto antibody reaction, and what antibody or immunoglobulin that is responsible. That is why we are studying autoimmunity in blood transfusion, and also to enable us to understand blood transfusion very well so that we can do safe transfusion whenever we are doing transfusion. Next slide. So what are the things that are causing autoimmunity? And the answer to the question is immunoglobulins. Immunoglobulins generally are antibodies. Another name for immunoglobulins is called antibody. And we all know that antibodies are produced by plasma cells, which basically are the B lymphocytes. So they are glycoprotein produced by plasma cells in response to antigenic stimuli. But antibodies that are produced are specific to a corresponding antigen. For instance, a particular antigen, for instance, let's talk about blood group now. Anti-A will produce, antigen A will produce anti-B. That is corresponding antibody for anti-A. It's just the same way when we are talking about immunoglobulin. Next slide, please. So this is talking about the structure of immunoglobulin. We are very familiar with the structure. It's like a Y shape, and it has two heavy chains and two light chains. So each antibody has a FAB region, which accommodates the antigen binding site, and also a FC region, where the complement activation, opsonization, and the ability to transfer through placenta are located. Next slide. So we're going to go to IgM, which is one of the most important immunoglobulins in the body. So it's a pentamer. What they mean by pentamer is that it contains 5 molecules, and the 5 molecules are joined together by what is called J bond. It's a very high molecular weight. That is, it's very heavy. So that makes it not to have power or ability to cross placenta. So the function is the first antibody that is produced during primary immune response. Is an excellent agglutinin because of the because of its large nature. So it can cause agglutination very fast, and it physics complement among the rest. So the clinical relevance is responsible for cold agglutinin reaction, which is usually found in ABO isoagglutinin, and it can be detected in acute infection. So IgM is present when the infection is happening, as in it is happening right now. IgM is the one that is doing the job. So let's go to another immunoglobulin, which is in IgG. Next slide, please. So the next two slides we'll be talking about, this one and the next slide we'll be talking about hemoglobin, their structure, function, and clinical relevance. For instance, IgG we are looking at now is a monomer, that is, it contains just one molecule. It's lightweight, which is the reason why it can cross placenta, and it is found both intravascularly and extravascularly. The function of IgG, it is the major antibody that is concerned with secondary immune response. That is after first exposure. Any other exposure, IgG is going to be the one responsible for immunity in that case. It has the ability to activate complement, though not as effective as IgM. Remember, the first slide say IgM can activate complement, but because it's not as heavy as IgM, its power is not, its activity is not as strong as that of IgM. So it helps in opsonization, which enhances phagocytosis. It neutralizes toxin and viruses. The relevance is seen in hemolytic disease of newborn, and also because of ability, and also is a warm autoantibody. Most warm autoantibody rather are typically IgG. It is used in indirect and direct Coombs test. That is, our Coombs test is an IgG based antibody. Next slide. So the next one is IgA, which is also an important antibody. This one is monomer in serum and dimer in secretion. That is, it comes in two phases. If it is found in serum, it comes as one molecule. But if it is found in secretion, it comes in two molecules. And the molecules are linked together by J chain and the secretory piece. Function: provide immunity in secretion like saliva, tear, breast milk, respiratory mucus. It prevents pathogen from adhering to mucosal surface, does not activate complement by classical pathway. What is the relevance of IgA? Selective IgA deficiency predisposes to mucosal infection. The presence of IgA antibody in IgA deficient individual can cause anaphylactic transfusion. Next slide. The next slide is just talking about the summary of all the three antibodies that we just discussed. So if you don't really want to go through these three slides, this one slide summarizes all the things we said in the previous three slides. That's why I just decided to make this table. For instance, for structure, IgM is pentamer, IgG monomer, and so on and so forth. The role is IgM is primary response, IgG secondary, just in case you are asked to make comparison between one and the other antibody. The next slide is the quiz. Next slide, please. So that's basically all we want to talk about, because I just decided to make the thing very brief and short. So the quiz is there. After looking at the slide, you can just look at the quiz and practice the question. So it will help us to understand the slide better. And I'm very sure that the topic is not alien to us. We all have come across the topic one way or the other. So we are going to go to the second lecture, which is on leukocyte and platelet antigen. Dr. Pan, the second slide, sir. Second slide, Dr. Pan.
Please, what's the second slide? What's the title?
The initial one that you said you.
Okay, the leukocytes, the HLA.
The leukocyte, platelet, antigen, and antibody.
Okay, please just give me a minute, please.
No problem, sir. Okay, thank you very much, sir. So this topic is a very broad topic, but I just decided to summarize it to maybe less than 20 slides, because like I said, we I don't want to burden us unnecessary. So let's go to the introduction. Apart from the red cell antigen, we all know that we have leukocyte and the platelets having antigen on their surfaces as well. So that is where this topic comes from. Leukocyte and platelet antigen and antibody. And of course, wherever you see antigen, there will always be a corresponding antibody. So on the leukocyte, we have antigen and we have corresponding antibody in the serum or the plasma. So also the platelets, there's antigen for the platelets on the surface of platelet and corresponding antigen as well in the system. So wherever we have antigen and antibody, and they come in contact with some other antigen antibody that they don't like, there is bound to be reaction, which is why we are asked to study this topic in blood transfusion, so that we will understand why some reactions are taking place during transfusion. So these antigens are important in transfusion medicine and also in transplantation immunology. Transplantation, we know, is putting one organ from donor to the recipient. Next slide, please. Basically, leukocyte antigen are divided into two major antigens. We have human leukocyte antigen and we have human neutrophil antigen. Leukocyte antigen is on all leukocytes. Why for neutrophil? It is specific for neutrophil. Next slide. So the definition of human leukocyte antigen system. HLA refers to the major histocompatibility complex in a human. It's a group of genes that is located on the chromosome 6 that code for protein expressed on the surface of nearly all nucleated cells. So this molecule presents antigenic peptide to immune cells and play a key role in immune recognition and self-tolerance. Next slide, please. So these are the classes of HLA molecule. We have class 1, we have class 2, and we have class 3. Examples are also on the slide, and what they do is also there as their function. For instance, class 1 belongs to all nucleated cells, and they present endogenous antigen to T cells. Examples are HLA A, B, and C. Class 2 belongs to B cell, macrophages, and dendritic cells, and they present exogenous. The example is in the front. So also class 3, which is the plasma protein. Example are complement 2, 4, TNF. Next slide. What is the significance or the importance of HLA? They are useful during transplantation because if you cannot match the donor organ properly with that of the recipient, there is bound to be organ rejection. So that is why we have to know we have to understand the HLA system in human being. Then they are also associated with certain diseases. For instance, the HLA B27 is said to be linked to ankylosing spondylitis, among others. Then transfusion reaction, of course, when there is a mismatch in HLA, at times there may be transfusion reaction. Then reproductive immunology as well. HLA is involved in reproductive immunology. I don't want to break down all these ones. It's going to make the notes more cumbersome. So next slide. So next slide is detection of HLA antigen and antibody. How do we detect them? Just like we detect antibody on the rest cell surfaces, we are supposed to understand how to detect all these antigen and antibodies too. So like this method now, microlymphocytotoxicity test is a way of doing that. Flow cytometry, ELISA,
Let me pause the
molecular
video.
method. Sorry? Hello, Dr. Pan. Okay. Detection of HLA antigen and antibodies. So microlymphocytotoxicity test. This test now is just like when we are doing blood group as well. Lymphocyte is mixed with certain things and then complement is added, then it is checked under microscope to check if there is dead lymphocyte. Dead lymphocyte will pick up the dye, while the live ones will not pick up the dye. By the time you start breaking this one down, it's going to make our notes very long and cumbersome. So let's just leave it as it is. But if you need to read by yourself, you can go ahead and read. But basically what it does is to detect if there is any antigen or antibody reaction in any lymphocyte. Flow cytometry, just like the one we use for our HIV CD4 counts. That's flow cytometry. Then ELISA, just the normal ELISA stuff. Molecular method, PCR-based stuff. Next slide. HNA. So we are talking about neutrophil now. So this one is specifically located on the surface of neutrophil, and they are glycoproteins and encoded in polymorphic genes. So the recognized HNA include HNA 1, 2, 3 to 5. Next slide. The importance of this is neonatal alloimmune neutropenia. For instance, if mother is secreting a kind of antibody that is against that of the baby inside, if the antibody escapes to the baby system, it's going to destroy the neutrophil of the baby, causing the baby to have neutropenia and exposing the baby to opportunistic infection. So autoimmune neutropenia, so that is self-induced destruction of neutrophils in the system. Then trally, that is, this one is when the recipient, we are talking about blood transfusion now, the donor and the recipient, when the recipient receive antibody that is not compatible with itself, I mean the neutrophil antibody, is going to destroy the lung. It will cause acute lung injury. That's the reason why we are studying HLA, HNA. Next slide. How can we detect this anti-HNA antibody? So these are the ways we can detect them: by doing GIFT, which is granulocyte immunofluorescence test, by doing granulocyte ablation test. These are all the methods we can use to detect these antibodies. Next slide. So we've moved to platelet now. We are done with the neutrophil and the leukocytes. So here is the platelet, just the same way. The platelet antigen are glycoproteins on platelet membrane, and they can stimulate production of allo or auto antibody. We all know what allo is. That's self. That's coming from outside. Auto is within. So when we are exposed to all this antibody, it could cause we can be exposed to this antibody through transfusion, pregnancy, and transplantation. Next slide. So platelet antigen can be classified into two: HLA class 1 antigen, human platelet antigen is the second one. Next class. Next slide. These are the importance of platelet antigen, medical importance of platelet antigen. Why do we need to do it? Next slide. Okay. How can we detect antiplatelet antibodies? The methods: by doing platelet immunofluorescence test, by monoclonal antibody mobilization of platelet antigen, flow cytometry, ELISA methods. These are the basic methods of detecting this antibody. Like I said, I don't want to break them down. For instance, this one is like when we are doing ICT. For instance, platelet immunofluorescence test is like doing ICT, where you mix the patient's plasma or serum with platelets, washed platelets, and then you now add something else. You will now add a reagent that will show whether it's agglutinated or not if there is antigen antibody reaction. This one also can be used for the detection of antiplatelet. All these methods are used. Next slide, please. So the application, okay, detector of antiplatelet antibody, we've done that. So the application of all this, the usefulness, that's what the application means. When we are doing donor recipient matching, during transfusion, for diagnosis and for research. So basically, those are the things that we can apply the detection to. So the last slide, I think it should be the last slide. Next slide, please. Okay, yeah, it's the last slide. So that's just the quiz. It's just for you to just study the slides and practice the question. So that is what we have this evening. That's all. I don't want to flog the course too much so that we can just have a grasp of it at the end of the day, because BGS is a very wide aspect of hematology. By the time we start breaking it down into pieces, it will become too voluminous for us. So thank you very much for being with me. I really appreciate your presence. If there's any question or any concern, you should bring it forward.
Okay. Thank you very much, sir. I'm very grateful. I want to ask a question, sir.
Okay.
Why is it that this HLA antigen, platelet antigen, all this, there are significant shows that we should include them in transfusion science, but in screening for blood donors, we don't do it. We just do normal cross matching without using this HLA method.
Actually,
Maybe if you
HLA.
are doing... Okay, sir.
Are you done?
Yes, sir. Yes, sir.
Yeah, HLA actually is for organ transplantation, because HLA is the tissue, like tissue transfusion. How would I put it? Tissue transplantation. For instance, if you want to transplant liver or any organ, you have to, there's no way you can homogenize part of the organ to get it, because we'll have destroyed the the donor's organ. But you can, from the blood, see the antibody from the plasma. because if the antigen is on the organ, the antibody will be in the plasma. So from the blood now, you can now do the matching using the plasma or the serum of both the donor and the recipient, so that you'll be sure that the organ that is being transplanted will not be rejected. Unlike the blood that you can easily is in liquid form, that you can easily do your ABO and the stuff like that. It is not possible for organ. Then again, ABO grouping is the most important for blood transfusion, although it's also important in HLA, because that's the first thing you have to do, because all the antigen and antibody in the blood cell are present in, except results anyway, are present on the surface of the organ. It's only results that antigen that is not present in other organs. So that is why we use HLA. HLA majorly is for tissue and not for blood. I don't know if I answer your question.
Thank you, sir.
Yeah. So if we have any other thing, can we just discuss it? Or if you have any addition to put.
Hello, sir.
Hello.
Sir, please, I have a question.
Okay, Abibat, right?
Yes, sir. Please,
Okay.
I want to know if these all these HLA, HNA can be detected through this normal cross-matching that we normally do for patients.
If HNA, HLA can be do what? Can be
detected.
detected.
Yes.
Using the
During
normal, using our
cross-matching. Yes.
using our normal cross-matching, it cannot be detected.
Okay.
It cannot be detected. For instance, if you want to detect for platelet antigen now, you have to use fluorescence dye, and it involves fluorescence microscope. So you cannot use our normal cross-matching to detect them.
And now I see, because after doing your normal cross-matching, they still report there is a reaction. You now begin to wonder, I've done the right thing, I've done it again. Okay.
You know, we have a lot of blood groups. There are some uncommon blood groups that can also cause reaction. For instance, like our Duffy, like our Care, there are sometimes that the titer of the antibody will be high enough to cause transfusion reaction. Then again, it might not really be blood that is causing the reaction. It might be maybe the drug reaction. And unfortunately, it now falls to maybe that time when blood is being given. That's why when there is transfusion reaction, you are asked to ask for another blood from the patient, the one that is collected immediately after the reaction occurs, where you are going to do a series of investigation in the lab, like you are going to do the DCT, the ICT, check the urine, do the bilirubin to know exactly where the reaction is coming from. Because not all blood transfusion reaction is due to antigen-antibody reaction. Some might even be microbial problem. For instance, if there is a bacteria in the blood, it might trigger reaction. And that might not be due to your ABO and resource incompatibility. Do you understand?
Yes, sir.
Okay.
Thank you, sir.
You're welcome. So any more contribution or query? So if there's none, maybe we should bring the class to a close. But you can always engage me on WhatsApp. My number is among those ones in class. So if there's anything you want me to
Yes, sir.
teach more,
We are very grateful, sir. Thank you very much, sir.
you're welcome. So thank you.
Yes,
Have a
sir.
nice week ahead.
Thank you very much, sir.
Bye.
Thank you, sir.
Bye, sir.
Dr. Akpan. Dr. Akpan.
Yeah, I was in class.
Okay, be on to now.
Okay. For those of us that are still in the class, please,
Yes, sir.
on the changes in the rate at which we are going to pay for the Zoom. It was yesterday that the class rep said it is no more 27,000 naira, and I asked him to let us know the actual rates for now. And today, he sent to me that it will be at 33,000 naira. Initially, it was 29,000 naira that he asked us to pay, but later he said we should pay 27,000 naira. I don't know if the changes is due to dollar rising. What he was trying to do before is that the link that we used to use for our lecture before, that was the one we have been using. But he said there are times we may have lecture in hematology, those in chem part or microbiology may still have lecture at the same time. So if we all have lecture at the same time, we cannot use just that single link. That is why he said it's better each department that we have our own Zoom link to be using. So it's not a matter of adding to the money. I paid the 27,000 naira directly to him even before everybody paid. I already transferred the 27,000 naira. Though everybody, as at now, everybody has paid for that 27,000 naira. Some even paid for 2 months, and some paid extra money. So we are just going to add 6,000 naira to the 27,000 naira that we have paid before. So we still have money in the account since some paid for 2 months. And later, if we now know if for the next month it is still going to be 33,000 naira, that means others will just add to, like those that have paid for 2 months now, they will just add token. And for those of us that just wait for a month, we are just going to pay the balance in addition to that one that we have in my account. So if there's anyone that has any question on it, please ask now so that I can ask Mr. Fasasi, and I'll get back to you at the WhatsApp group. Hello.
Hi.
Hello.
Hello, Fatima.
Hello.
Hello,
If you
hello, I'm
can hear
hearing
me, calculate whatever is remaining and let us know.
you. All right, ma'am.
Hello, class
How
rep,
much
ma.
are we going to pay
Hello,
moving
sir.
forward? Hello, ma. How much are we paying moving forward? You know, everybody, like you said, I've already paid 1 month. Some have paid more than a month. So maybe subsequently, how much are we paying? Let us know how much. You know, before you said it's 1,000, I think 6 something.
1,675 naira
So,
50 kobo.
so maybe how much are we paying subsequently so that the class will just prepare for it.
So I will still discuss with class rep that is the 33,000 going to be constant? If not, you should let us know what we are to do in subsequent months. And for those that have paid, our private tax individual, since we didn't pay the same amount, some paid 2,000, some paid 2,001, some paid 3,000 naira. I will send to individual based on the balance that remains to meet up for 2 months.
All right, ma. Then in addition, ma, please, the first payment, has that one expired in the month of
We have not even used
October?
it. We have
Okay,
not
I've not
even
used it.
he said he's there to even pay that one. We have not even used that one.
Okay. So that our payment now, what I'm just asking is, when does it take effect? The monthly payment, the one
That
we have
was
used. Okay, he said he has
that
not.
was
Okay.
that was what I asked him yesterday. That when is the first month starting and ending? so that I will know the time we are going to pay for the second month. That was the time he now
exactly.
said we have not even used the money, that he has not even paid it at all because
That
the
means you
rate
have been
has
using
changed.
bonus. You have been using bonus with that now. Okay. So
Yes,
if you just
yes. Maybe from
if,
the previous
if
ones that we have paid for the first
okay,
session.
if you just let us know the specific time one is starting and the time it is ending, so that at least we'll be prepared. We'll prepare ahead. Shema, thank you so much,
Yes,
ma.
sir. All right. Thank you very much, sir. Please, though, if anybody has comments or observation or any reaction, please now say your mind. Okay. Thank you, everybody. So I will assume that we are all okay with it.
On the same page.
All right, sir. Thank you very much. Bye, sir.